What I want to do today is a little different than my normal scientific lectures, and Philippe had said this would also be open to the public. So I want to discuss the concept. We'll discuss a little bit about the science involved of how we understand how these microbes work, but I think it's very important to also stress that as we understand these processes, we can actually use this knowledge to our advantage and develop things that might actually counter these organisms. And so that's the theme of today's lecture. The other three lectures will be much more detailed and scientific as we go along. But today I want to sort of talk about things that maybe even Philippe hasn't heard me talk about that we've done. So I want to go way back to this very famous quote. U.S. Surgeon General, big fellow in the U.S., basically said, "It's time to close the book on infectious diseases. We are done. We don't need to study this anymore. We have antibiotics. We have vaccines. Let's move on to something challenging like cancer." And we all know what happened then. Each of these little red dots are various infectious diseases outbreaks. You don't have to see them all. But it's really not time to close the books on infectious disease. And I don't need to talk to you too much about that, but I think you realize infectious diseases still are a major problem in our societies. So the nice thing about infectious diseases is they really do not know borders. They're worldwide. They're global. And you can have an outbreak somewhere in some place in the world, and within 24 hours, it's elsewhere in the world. So that's neat. I get to come to France, and I see Philippe all over the world. It really is global in that sense, and also from the infectious diseases. So I kind of want to start the story just to emphasize this. This is a case a while ago where a person got on a plane in Chicago. They had multi-drug resistant tuberculosis. And they then sat down in this seat right here, and then they flew to Hawaii. And that's a very long flight, about 8-10 hours. And during this time, they did what people with tuberculosis do. They cough and et cetera. And because they knew where this person sat, they could actually then realize who all this person had, basically seroconverted, who else had raised antibodies to this. And that's what all these other seats represent, people that actually seroconverted due to their exposure to this patient. This is a flight attendant here. So, I always think that, this is a reminder that, no matter where you are in the world, you can always be potentially exposed to infectious diseases. And then also jokingly say that if you're ever on an airplane, try and sit up at the front and upgrade to business class because it's much more comfortable than sitting at the back. Unfortunately, she did die of the disease. Fortunately, the others did not go on to fulminant tuberculosis, so they just seroconverted and that was it. So I think we all generally sort of hear about these things, just the numbers of infectious diseases in our society, and you can see that, greater than a quarter of all people worldwide still die of an infectious disease. It's phenomenal numbers of people die. 14.9 people of the 54 million people worldwide. And your first reaction is, oh, that's just often developing countries due to HIV, malaria, TB, et cetera. But it's actually not so true. There's actually many other diseases impact in our societies, developed societies, as well as the developing world issues. The other problem compounding this is we've actually been very poor at coming up with ways of combating these infectious diseases. We really don't know how to actually go at them. Can we actually treat them? Can we come up with things? And this is to say that we all know that antibiotic-resistant infections are continually rising. These are, for example, methicillin-resistant Staphylococcus aureus, which is an organism that's become resistant to most drugs, and other organisms are also resistant. These are going up, and they continue to go up. So in the U.S., for example, over 60% of all Staphylococcus aureus infections, which is a common infection, are multi-drug resistant. Well, that would be great if we had more drugs to throw at these things, but we don't. And this is the number of drugs that are being approved by the U.S. FDA over the years. And you can see that this is just plummeting. There's very, very few new drugs that come on the market. And the few that do come on the market are really just a derivation of previous drugs, so the bacteria become resistant very quickly. So we really have to do something different. And that's kind of the concept I want to probe today, is that if we start to understand how these things work, maybe we can use that knowledge to come up with new ways to combat an obviously ever-increasing problem in our society. So Joshua Lederberg is a very famous microbiologist, Nobel laureate, and he came up with, I thought, was a wonderful quote, much better than the U.S. Surgeon General's quote, really, that, it's our wits, our smarts, our ability to figure these things out versus their genes. we are not going to multiply the bacteria. They can multiply in 20 minutes. It takes us 25 years to divide. So we can't out-divide them. We have to use our heads and understand how they work and come up with ways of combating these things. So I'm going to we'll sort of just take a cursory view at some of the organisms we work on and then embrace that with some of the science that we found about them to then lead to actually how these things work. And so we'll start off with Salmonella. This is an organism we've been studying for many, many years. And in people, it causes two types of main infection. It causes typhoid fever, a very serious systemic disease. You see, especially in developing countries, you can see the numbers here, very, very large numbers of people. And then there's gastroenteritis. This is nausea, vomiting, diarrhea, a very, very common disease worldwide. You can see the numbers, including our societies, phenomenal numbers. And the other problem, like with many infectious diseases, is we're seeing increasing in drug resistance. Salmonella is becoming more and more drug resistant. So I'm not going to say a lot about Salmonella. We'll have a whole big lecture on it near the end of the month, but I will just remind you generally what it does. So when you swallow Salmonella in contaminated food or water, it goes down to the intestinal epithelium where it then binds to the cell surface or somehow attaches. It then triggers a massive event in the epithelial cell wall that basically causes the engulfment of the organism. The bacteria then goes in and is surrounded inside the intestinal epithelial cells by these membrane-bound vacuoles, and it's now inside these cells. So it's now an intracellular organism. And it uses this way to actually penetrate across the gut barrier, across the intestinal barrier, to then get into the body. So this is an area that's been studied by our lab and many others over the years a lot. And really what we understand is Salmonella has a very specific way of driving bacterial proteins into host cells, this thing called a type 3 secretion system that basically is a syringe to inject bacterial molecules into host cells. Once these bacterial molecules are inside, they do spectacular events to the cytoskeleton, things like actin inside these cells, and you can see the bacterium here and bacterium at the center of this big, we call it a splash event. It's kind of like throwing a rock in the water. You see the splash of the cell surface, then come up and embrace the bacterium, and then drag it in. And if you stain the cytoskeleton, the actin, you can see it's centered around these yellow bacteria. All the red stuff here is the actin around the yellow bacteria here. So many labs have made careers out of understanding what these yellow bacterial molecules are that are inserted in the host cell. I'm not going to say much about these today, other than the end result is they really do drastically reprogram the host cell's actin cytoskeleton to allow it to then take the bacterium up. So this is just an animation that kind of summarizes this first event of Salmonella. It comes along, and then it will have this type 3 secretion system called the SPY1 that is then inserted into the cell. It then drives bacterial molecules in the cell, which then trigger the engulfment of the bacterium. And that's great, but now the bacterium's inside, and it has to now figure out what to do once it's inside this cell. So this is when it actually it has another second secretion system, this thing called SPY2, another hypodermic syringe that injects more bacterial molecules into the host cell, and these things basically set up a protective environment in which the bacterium then lives. So it's going to coat this vacuole with these bacterial proteins and others that basically forms a protective environment. So if you know anything about cell biology, you know that when things normally go into a vacuole, they fuse with lysosomes, which degrade the bacteria. Salmonella has developed these things that basically are protective mechanisms, so the bacteria then doesn't do this. So we're just going to pick this up further. We're now inside the cell looking up. Here's the actin filament Salmonella. You will see it come in here. So it then drags the bacterium into this cell here, and it then forms a little protective environment here. So you'll see the vacuole here. These are things called early endosomes. They fuse with the vacuole. That's okay. That's not bad for the bacterium. And the bacteria then gets really sophisticated and starts putting out the second type 3 system, the SPY2 system, which then forms a protection around the vacuole. And the bacteria can then begin to safely divide in this environment. So that when lysosomes come in, they then bounce off, and the bacterium sets up a stable environment in there. It even has the capacity to actually redirect the formation of this thing. It will sprout some tubules here. We'll talk about these in a moment, that are somehow key for the life of the bacterium in here. So it then starts to divide in here, and the vacuole gets more and more spacious, and the bacteria then swim around inside this thing. How it gets out further from there to infect others, we don't actually know the details of this yet. So it makes all these different bacterial molecules that are then delivered to the cell that do all sorts of things in terms of reprogramming the cell, so this does not basically degrade the bacteria. Okay, well, we know these things, so the question becomes, well, what can we do about it, and how can we actually start to think about countering, say, salmonella? So one of the ideas we had goes back to the early days of when microarrays became available. We could suddenly look at what genes were being expressed inside a cell in response to infection. And the other thing overlaying this was the discovery of things called toll-like receptors, or TLRs, which seem to be key for the importance of basically maintaining antimicrobial activities in the cells. So this is called innate immunity, and it was kind of being rediscovered starting in late 1990s, for example. And what we realized about innate immunity is it's the reason why you and I are basically not sick every day. It's a very efficient system, and it deals with most of the microbes that you encounter pretty much all the time. Rarely does it not work, but if it doesn't work, then we have the acquired immunity, T and B cells come in. But these things only come in at least after a week. So what's kept you alive for that week is the innate immune system. So this is a very efficient defense system. And the idea went that, well, okay, if it's so efficient, if we could find handles of this system, we can actually enhance these innate responses with, say, compounds, and then these things actually should alter the outcome of infection. So to make a long story short, we used genomics and arrays and looked at host response to infection and started to map pathways involved in these things. And we then spun off a biotech company that allowed us to identify lead compounds that basically would treat infections in mice, but they wouldn't kill the bacteria. They're going to basically enhance the host resistance. So they're not going to directly kill the bacteria like antibiotics would, for example. They're instead enhancing the host resistant mechanisms to then counter this activity. And in theory, they should be fairly broad spectrum. They should work against many different organisms because it's a sort of a generic defense mechanism instead of a very specialized defense mechanism. So the company has gone along very well, and we started to make different compounds, and these are the number of salmonella. These are counts of salmonella inside the animal. You can see they decrease with different compounds, that we can actually start to counter the number of salmonella in a cell. And the theory of, well, it should work against many different bacteria, we also put in Staphylococcus aureus, which is a very different organism than salmonella, gram-positive organism compared to gram-negative. And again, it had significant drops in the counts of Staphylococcus aureus in these cells. And yet, if you mix the peptides with salmonella or Staph aureus, they wouldn't kill the organism. So they're enhancing the innate responses to this. So if the idea being that work differently than antibiotics, in theory, the effect should be additive. We should be able to give antibiotics, get an antibiotic effect, add this peptide, get another effect. Here's the antibiotics here, here's the peptide here in terms of total counts. You put the two together, it is additive, and we then take the organisms down to an even lower level inside this infection model. So this has gone extremely well. The company is called Inamex, and we've now gone through, I think, $42 million in venture capital funds. The peptide has now gone through phase 1 trials, and it's about to enter phase 2 trials now, and we're very hopeful that it would be a new way or a new approach of treating infections, instead of making direct antibacterials, enhancing the host innate defenses to this. Well, that then brings up this fellow here. This is Bill Gates, and he basically said, well, maybe his wife said, you know, we've got all this money, we made all this money in computers, maybe we can actually do something to help humanity. And he came up with what was called the Gates Grand Challenges. And it's based on a principle developed by a mathematician, David Hilbert, where he basically said, here's all the major problems in mathematics that have not been solved at 1901. He basically posted them and challenged mathematicians to then go and try and solve these things. And over the years, they picked these things off, and they did solve them all. And so they've actually all been solved in this time. So the Gates concept was the idea to take a fair bit of money and challenge scientists in the world to basically come up with science and technology approaches to basically enhance life in the world, especially through the progress of diseases in the developing world. So it was a fascinating program, and there were many, many applications, and we were involved in one, and Philippe was part of this team to do this, and this is sort of to capitalize again on the work we did with the innate immune modulators to then develop other compounds we can use in developing countries to counter infectious agents. So this is the grant that was awarded, a fair bit of money, and the idea was to really come up with a program that we could actually use to come up with compounds at the end of the program to work in this. So the idea was to really measure major responses of the innate pathway and also how the immune modulators work, form this massive data bank, and once we understood the data bank, we could then use this knowledge to generate lead compounds and lead to delivery. In reality, it actually went a little bit differently than that, in that we got these compounds and everything before we actually fully had the data bank formed, but the data bank has now been extremely useful to do this, but we did do what we said we were going to do at the end of it. And if any of you work on innate responses, etc., I highly encourage you to have a look at this database. This is a massive database of all the interactions that occur during innate immunity, and it's a fabulous resource that's on the web for understanding innate immunity. So I'm not going to go through all the work that went on in the various things. I just want to tell you about one thing that's now come out. It's been very promising, and this is against a parasite, malaria. Now, it's not the parasitic burden that kills so many children with malaria. It's basically the cerebral inflammation that kills these kids. And there is mouse models to this. And as part of this program, we were testing these various immune modulators in various models. And with Louis Schofield in Australia, we found that these innate modulators could actually have a drastic effect on mouse survival due to cerebral malaria. So normally in cerebral malaria in these mice, all the animals die at all times here. Yet if we actually use these compounds, you can see that they, in this case, gave us 40% survival. Many other compounds are 60–70% survival. And this compound is now going forward in Australia, moving towards clinical trials for cerebral malaria, because it's an extremely difficult disease to treat, and like I say, it's the cause of death in children. So another idea of the enhancing innate responses, this also dampens the harmful inflammation associated with malaria, and seems to be very effective, at least in this particular mouse model. So I'm now going to switch to another organism we study a lot about, and this is pathogenic E. coli, and Philippe introduced this in the introduction. And just as a reminder to you that there's many kinds of E. coli's. There's those that are harmless and we use in the lab all the time to clone with, yet there's many others that cause disease in people. They can cause urinary tract infections, meningitis, and as well as diarrhea. The ones I'm focusing on are those that cause diarrhea, especially in young kids in developing countries. We don't see it so much in developed countries. There's the odd outbreak here and there, but it's not that big a problem in our society. But in developing countries, it's a big problem. And the hallmark of this organism, others related to it, is they can actually bind to intestinal cells. Instead of going into cells like Salmonella, they then live up on the surface and then live there, living on what we call these pedestals, and they just live up there. So there is a close relative of E. coli, EPEC, and this is something called enterohemorrhagic E. coli, EHEC. And this is one that we do see, especially in North America, not in France so much, but in North America we see a lot of it. And it's caused by cows carrying this organism, but the cows don't get sick. They carry this organism, they secrete it in their feces and contaminate water, or when you grind up cows to make hamburger, you then get what's called hamburger disease because it contaminates the hamburger. This disease is a watery diarrhea, which then progresses to a very bloody diarrhea in both children and, well, people of all ages. And in young kids, they have a really terrible kidney disease called hemolytic uremic syndrome, because this bacteria makes this thing, this Shiga toxin, which then goes around poisoning cells, and including kidney cells, and leads to renal failure and either death or dialysis of these things. So it is a very close relative of the enteropathogenic E. coli, and their virulence mechanisms are virtually the same. So there was a major outbreak in North America a couple of years ago now, where there was a big outbreak in spinach, and it was because the cows basically had defecated. There was fecal contamination in the water. The water was used to water the spinach in California, and there was a major spinach recall. So if we take our host intestinal cell and we add our E. coli to it, you basically get diarrhea. And so the question is, well, how do we actually do this? So if you go looking in samples of stool over the years, you won't find O157 pre-late 1970s. It just wasn't there. The organism doesn't exist. It basically arose out of events in the bacterial world and really a series of genetic events. And I liken to say that the bacteria in the gut, it's kind of like this genetic internet where you can get any gene you want to from your neighbors. You can get antibiotic resistant genes, virulence genes, et cetera, et cetera. And when you line up the E. coli in the K12 lab strain versus the O157, basically there's about 20% DNA differences as well as the 80% that says I am E. coli. And there's one region that we've been focusing in for years now that basically contains most of the genes needed to do all the disease aspects of the O157. So what we think happened is that we had an interpathogenic E. coli-like organism which had this genetic components in it. We then add the Shiga toxin, which is encoded on virus, to the organism, and you put these two things together, you then get a new pathogen, which is the E. coli 157 that we see today. So how does E. coli work? Well, like Salmonella, it's got a whole set of events it uses to cause disease. It's got something called a bundle-forming pilus. This is basically a grappling hook that allows the organisms to initially hang on to the intestinal epithelium as it's going through the intestinal tract. And it comes along here, and it then adheres to the cell surface. It triggers a massive retrenchment of the microvilli intestinal surface, and then it then starts to settle down. And getting back to that region of that genetic region I was telling you about, so it's about 1% of the chromosome in terms of size. And what does it encode? Like Salmonella, it encodes these type 3 secretion systems. We see these in many, many gram-negative organisms, which gives the bacterium a wonderful ability, at least from their point of view, to derive bacterial molecules into host cells. So about 2/3 of this is dedicated to secreting molecules out of the bacterium. Then there's this region here that is things that get secreted out of the E. coli into the host cell, these things called E. coli secreted proteins. And then there's a region in here that is needed for really intimate contact, and I'll say a bit more about that in a moment. So this big genetic chunk here is really pretty much all you need to form those pedestals on cells. So we've learned a lot about this system over the years. Again, I don't want to go into detail of it. It uses energies to basically drive secreted molecules. The chaperones dock them here through a very complex system here to get it across the bacterial inner membrane and then the outer membrane and then down into the mammalian cell. And over the years, our lab and many others have sorted the details of this out, and we know quite well now generally what actually happens in terms of molecules that are being secreted. They have chaperones that bind, docks them, ATP drives them through here, and then there's these proteins here that are actually involved in penetrating into the mammalian cell, and then basically you then have a conduit directly into the mammalian cell here. And when Philippe referred to earlier, we discovered many years ago the ability of the bacterium to drive a bacterial molecule, this thing called TIR, into the host cell, where it becomes in the host membrane, and it then reaches up to then bind intimin on the bacterial surface, which is a non-membrane protein, and then locks the bacterium together. So it injects its own receptor into these things. It also injects several other proteins. You can see some of these things here that are involved in various cytoskeletal aspects to the whole process. So I've already mentioned this molecule here. This is the bacterial molecule that goes into the host membrane. Part of it reaches up to bind intimin on the bacterial surface, so this intimin binding domain. And then the other part of it sticks down in the cytoplasm. It even has tyrosines that gets phosphorylated. And this region here is absolutely critical for nucleating the actin to then drive the pedestal onto the bacterium. So we solved the crystal structure of this thing a few years ago, and we certainly know at a molecular level what actually occurs here, these interactions, and we know this binding is absolutely critical for the bacterium to bind to the host cell. So the bacterium binds on the cell surface. It then has this type 3 secretion system that forms a bridge down into the intestinal cell surface. Here's intimin up here that it will eventually adhere to. It then drives in molecules, things called ESP B and D, that form a pore in the host cell. And then you'll see TIR come along here in red that will then come through, I think. And then TIR, we think it actually drives directly into the bacteria, into the host membrane. It doesn't spend any time in the cytoplasm. It gets tyrosine phosphorylated that then nucleates the actin, etc. Then once it's embedded in the outer membrane, it's in the membrane, it then faces upwards, and then it reaches up and grabs the bacterial cell surface. This type 3 will then retract, and tear will then bind intimate to then lock the bacterium down. And then the other end then starts nucleate actin, and this is what then starts to basically form the pedestal on the organism. So this then nucleates into the bacterium and then forms this big actin cloud on the bacterium. It pushes the bacterium on the surface, and then that's where we get our pedestal from. So we spent a lot of years trying to understand the cytoskeletal components, and again, not in this lecture, I'm not going to go through this, but we really have started to identify both the bacterial and the host components that are involved in forming this actin pedestal to form this large protrusion under this organism. And we know in great detail now how this all works, and basically this phosphotyrosine, at least in EPAC, can then bind this SH2 adapter protein, which binds this complex here. And this is the complex that drives the actin to actually form these pedestals under the organism. So I didn't want to get into too much detail about how E. coli does this, but I do want to say, okay, so we've got this E. coli, got this complex way of adhering to cells. What can we do to actually think about doing things to counteract this organism? So just to remind you that really there's no current therapies about E. coli. If you add antibiotics, you actually make the disease worse because you release more Shiga toxin, you actually worsen the disease. And cows are the big carrier in this thing. They don't show any disease symptoms, but they up of half and even up to 70% of all cows now are thought to carry this organism. So they carry it, and it's the cows causing the contamination that leads to the human problem, either through hamburger contamination at slaughter or through contaminated water that leads to agricultural product contamination, such as Japan. They fertilized fields of horseradish sprouts, which are then used in children's lunches, and they had a major outbreak there. Or in the west coast of the United States, they used unpasteurized apple juice, which caused these problems. So instead of developing a human vaccine, the concept was, and I'd been thinking about a human vaccine for a long time for children, and actually I was just out running one day with the idea came to me that we should vaccinate the cows against this problem, because if the cows didn't have 0157, it wouldn't then spread to the humans, and maybe we could block the contamination of this thing. So this is a humorous slide that really shows what I know about cows, and I didn't know a lot, so I turned to a veterinarian that worked with cows, a guy named Andy Potter. And the idea was, I said, Andy, can we vaccinate cows for all these proteins I've just told you about here, and then they could make basically protective antibodies to these things so that the cows swallow the bacterium couldn't stick and would flush out the other end of the cows. And this concept actually turned out to work beautifully. It worked terrifically. And this is the first experiment we did. We took cows. We gave them basically a big bolus of 0157 orally. And we just tracked the number of days the cows shed 0157 in the feces. And in the placebo, like not vaccinated, the control vaccinated, basically they shed for 4.5 days. So E. coli colonized and shed for a while. If we vaccinated with the supernatant from the wild type, we just basically had a 0157 coming in. First little bit, they came out the other end, and that was it. They were gone. So the vaccine did work. We also tried the supernatant from a tear mutant. I told you tear was so important in adherence, thinking it was critical for it. It turns out that there's many other components besides tear that contribute to it. Tear obviously plays a role, but there's many other proteins in the vaccine that are also helping give protection to this. So this has gone extremely well. It's gone through clinical trials in cows, which is basically feedlots in both the United States and Canada. It's gone through about 60,000 cows now. It works beautifully. It takes 0157 to levels that are below the levels of 0157 you see in the winter when you don't have any 0157. And it's being used to vaccinate cows in Canada. And it's pending in terms of the United States. And I was just at an opening a little while ago where this company has now just built a 5,000 liter fermenter to produce this vaccine. And there's many countries, for example, Sweden are looking at adopting this as well, and the UK where they have problems in petting zoos with 0157. And it's really the only vaccine that seems to be doing anything in terms of decreasing the amount of E. coli. So there's another example. If you go through all the problems and figure out the science, you can actually come up with something, in this case a vaccine for cows. It's been a very interesting process because there's all sorts of other things that one doesn't think about besides the science. Like, who's going to pay for this vaccine? The cows aren't getting sick. So a farmer says, well, why should I pay for a vaccine for my cows? My cows aren't sick. Yet in reality, it causes major problems. So ironically, it looks like it will be the meat producers that will be involved in doing this, because that will avoid all the major recalls of contaminated beef that we have all the time in North America. Okay, I want to finish up on a viral disease, and something that I got into almost by accident, and tell you a bit about SARS. So I don't know if you remember, way back in November 2002, there was this scary stuff circling around the world, that there was this awful, weird flu-like illness occurring in China. And the first reaction was, it's pandemic flu. And we know what pandemic flu has done historically, and really they were very scared that it was doing that. There's all sorts of interesting rumors floating around the world, including it was a genetically engineered Ebola virus, and many other rumors that were out there. We just didn't know what it was. And as you know, it then went to be spread rapidly around the world, especially in Canada, but also around the world, and huge cost economically to the world, as well as some deaths. So Canada really was hard hit by SARS. There's a large Asian connection in Canada, and we have a very strong healthcare system. You can see the number of cases we had and the number of deaths we had. And ironically, we had the highest case fatality of healthcare workers, because we have such a good healthcare system. People coming in, they were being intubated. These nurses and doctors were in close contact with these patients. And nearly half the fatalities were actually doctors and nurses in this case. There was huge travel advisories. Don't come to Canada. It has SARS, even though it was in Toronto, which was 3,000 miles away from where we were, for example. And it had big economic implications. And there was a lot of fear around the world. We didn't know what it was. We don't know what it was caused, and what was actually happening. So there was a genome sequencing center in BC set up. That's the province I'm in. It's kind of like a state in Canada. And it was designed to sequence things. And one of the things they'd done is write a research proposal to the U.S. To combat bioterrorism, saying that if there's a bioterrorism agent, we would come up with a really neat way of rapidly sequencing this thing. Now, that grant was rejected for whatever reason, but they had this mechanism in place to sequence a virus as fast as you can. And so when SARS came along, they said, well, let's try sequencing the SARS vaccine that way. They applied that system, and it worked beautifully. They were the first people in the world to sequence the SARS vaccine. They beat the U.S. CDC by a few hours. They beat Hong Kong by a day, and the U.S. and Singapore by 3 days. So great, we were suddenly the first in the world to sequence the SARS virus. What are we going to do with this? Well, there was all sorts of discussion, what can one do with it? And originally, the idea was just put it out there so everyone could use it and use that information for the good of humankind. But then other countries started patenting things with SARS, and they said, well, no, let's not do that. We were the first one to do it. So then, retrospectively, they applied patents to it, and the idea was to let anyone use the patents and the protection for whatever they wanted to do, but not be able to make money from it. So we had the sequence of SARS, but really didn't have anything to combat SARS at that time. And so the idea was to discuss, well, what could we do to it? Could we come up with a drug or a vaccine should SARS come around the next flu season? And that's where I came into the picture, and our provincial government had some money they wanted to say, and let's see if we can't develop what we call the SARS Accelerated Vaccine Initiative, or SAVI, to come up with an alternate approach to actually come up with a vaccine in a real hurry, using all the expertise we had around the country to this. So the idea was to use this money to do what we called rapid response science, get people working in expertise areas we needed to basically use their expertise towards developing a SARS vaccine, all the different stages are needed to run this vaccine, and see if we can actually come up with a vaccine in a real hurry, because we were scared. We have a provincial funding agency that basically handles the funding of health research money. This money was put under them to manage it, so they were making sure we didn't spend it all on alcohol and nice dinners and things. And so what I implemented was, the first thing that slows people down doing science is how long it takes to write a grant, review a grant, and then get funded. That takes like 18 months. We don't have that kind of time when we're trying to have a vaccine ready in under a year. So I conscripted some peers I knew, some really good virologists around the country, 4 others. And the idea is that if you had an idea that could help develop a vaccine, it had to meet 2 criteria. good science, but also had to help us get the vaccine and be a needed area. So you would send me a 2-page PDF of this proposal. I would send it to these guys by email overnight. By the next morning, we would have made a decision and the money was in their lap. So we had a 24-hour turnaround for funding of this thing. So there we just saved ourselves 18 months. And we applied these kind of strategies wherever we could. The other thing we did is instead of doing one thing at a time like we usually do in science, that's, we do this, then we do this and do this. We mapped out all that we needed to do in the whole thing, in the whole vaccine production, and started everything at once. So that when we needed, say for example, tissue culture assay, neutralization has to be done, that will all be worked out. When we needed an ELISA to be done for, say, some vaccine response, that would be done. When we needed clinical trials to be done, we would start to have those things in places. So we did all that at the same time, and that turned out to be a profound way of moving things along very rapidly. So it was people all across the country, and really when you think about how we do research, we don't normally do it that way, but we needed to because we had to get something in a real hurry. The reason I got involved, and I thought it was a really neat exercise, if we figured this out, we could do this in any emerging infectious disease, and we all know there's more diseases coming. So we didn't know what kind of a vaccine to make, so we thought, well, let's try all the approaches of vaccines. We could do whole killed virus, we can do recombinant pieces of virus, or we can do live attenuated virus. So we did all those three in parallel, and we're the only people in the world to do them in parallel. Everyone else just chose one, for example. There was also lots of other things besides science that go into this whole thing, and things we really didn't appreciate. The media calls you every day and say, how's the vaccine coming? There's all sorts of regulatory issues. If you're using a vector to make this vaccine, where did this vector, that's patented by somebody. There's intellectual property issues, etc. And we really wanted to get this thing as fast as we could into relevant animal models so we could then go to humans as quickly as we need to. Luckily, this thing worked just beautifully. We really led the world in this whole thing. I remember being at the WHO a few months into it, and one of the things we set up was this interactive website that everyone could use, and pretty much every talk at this meeting was using our website, our bioinformatics site we'd set up, and they were using many of our slides. And it was really gratifying because many people, including the US CDC, said, well, we're going to do this, this, and this. And we actually stood up and said, well, we've actually done this, 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 and this. And it was really a neat time. So within 6 months, we're already testing in 2 relevant animal models. If you can prove it works in relevant animal models, if it's a really awful disease, you can actually get approval for that vaccine without actually doing it in humans if it's warranted, if the sky is falling and really millions of people are dying. So we did that in that pace. So within 1 year, we completed all our animal testing. We were ready to go. And really, it was an astounding application of real science. Boy, did we learn a lot of lessons. There's all sorts of lessons involved. One thing is that this is a real threat in Canada. It was kind of like a national, you're kind of being attacked, even though it was a biological agent. And everyone was really willing to help and provide their expertise to it. So people were really willing to help. It was a real imminent threat. You have to use people's existing expertise. You can't go and retrain a whole group of people to do something new. They have to already be doing it, but just switch to what they're doing it on, SARS, instead of whatever else they're working on. So the idea is using existing expertise. If any of you are lawyers in the audience, I apologize. Lawyers were by far the hardest thing to deal with. There's all sorts of intellectual property issues, and this was probably our biggest failure. We could not figure out how to get around all the intellectual property issues despite all this. So finally, we just gave up and we just did the vaccine, and we made enough to vaccinate people in BC if we needed it, and we figured that if so many people were dying by the next year, the government would figure out how to get around these lawyers, because that we couldn't get around. There's all sorts of issues. Who's going to produce this thing? Where the production facility is a level 3 organism, for example, and there was much national, international coordination needed. So all sorts of many non-science issues. Even once you solved the science issue and showed the vaccine worked, there was all sorts of other things that went into this. So a question you often get is, will SARS come back? My guess is not as we knew it. There's been a few lab-acquired infections, but SARS has basically disappeared. We know too much about it now. We know how it's transmitted. We know how it's how to contain it, how to prevent infections, et cetera, et cetera. So, and there's all sorts of interesting ways already put into practice in emergency rooms preventing these kind of things. So I don't think we're going to see SARS, but you know there's going to be something else. If it's not SARS, it's always something else. So that sort of brings me pretty much to the end of what I wanted to say, and people have said, well, okay, those are interesting, what are you doing now? This is just a preliminary glimpse of some of the data we're getting with a really neat new salmonella vaccine we're developing. So salmonella, I told you, causes typhoid fever and gastroenteritis. There are typhoid fever vaccines around, and these work pretty well around the world, so there's no need for a typhoid fever vaccine. What there is a real need for is what's called non-typhoidal salmonella. So this is the gastroenteritis type thing. And for example, in Malawi in Africa, 50% of the children that have non-typhoidal salmonella that have HIV die. So it kills half of all kids in Africa that have HIV that gets this disease. And this is a very common disease. And there's nothing you can do about it because these are multi-drug resistant organisms. So we've stumbled into a way that we think is actually working. And this is just the two mouse models we use all the time. It's not gone into humans yet of looking at it. And it turns out it seems to be working spectacularly. This is a log scale here. So we're getting basically at least 9 logs difference when you vaccinate animals with this vaccine in the typhoid model and also in the gastroenteritis model, which is the non-typhoidal model. We're seeing basically sterility when you vaccinate with this is at least a 7 log difference, which is just unheard of in the vaccine world. So we're very excited that we can actually start to develop this as a way of going forward for the non-typhoidal salmonellas, especially in developing countries or travelers going to these places where these multi-drug resistant salmonella exist. So this is really a thank you to the many people in the lab over the years that contributed to all this work. We are a big group. This is actually a picture taken in a lab retreat up in Whistler, which is where some of the Winter Olympics were held. It's about an hour and a half out of Vancouver. And I'm not going to go into the names of all the people who contributed to this, but there's been many, many wonderful people over the years that have been involved in this. And many have gone on to have their own scientific careers around the world. And really, it's just a warm and heartfelt thank you for all their incredible hard work over the years in all these different systems. So with that, I remind us that no matter how much we knew, maybe these bacteria will always have the last laugh. And I think that takes me to the end. Merci beaucoup.